Let's take a minute and summarize what we've discussed so far about the photosystems. Each of the photosystems has a group of chlorophyll molecules that serve as the antenna complex and a group of chlorophyll that serve as the reaction center. Remember, all a photosystem is a group of between 200 and 300 chlorophyll molecules that function together, part as the antenna complex and part as the reaction center. And there are two photosystems, Photosystem 2 and Photosystem 1. The electrons that reach the reaction centers from the antenna complex get transferred in the reaction center to an electron acceptor molecule. The next thing that's going to happen is that electron acceptor molecule is going to get oxidized. It's going to donate those electrons to an electron transport chain. The electron transport chain that is present in the thylakoid membrane is very similar to the electron transport chain that we saw in the inner mitochondrial membrane. All the electron transport chain is, if you recall, is a group of proteins. They're specially structured to range in electronegativity, and that allows these groups of proteins to pass electrons one to the next and harvest energy from those electrons along the way. So let's start with photosystem 2. Remember, photosystem 2 is actually the first photosystem in the process of photosynthesis. The reason that we don't call the first one photosystem 1 is because photosystem 2 was actually the second photosystem that was investigated in the laboratory. The first photosystem that was investigated in the laboratory was the one that got the name Photosystem 1. So yes, even though the names would suggest otherwise, Photosystem 2 is the first photosystem that is involved in the process of photosynthesis, followed by Photosystem 1. Photosystem 2 is going to harvest energy from photons of light from the sun and pass that energy along in the antenna complex to the reaction center. That reaction center will then transfer electrons to an electron acceptor molecule, which will then donate those electrons to an electron transport chain. Now, just like we saw in cellular respiration, in photosystem 2, the electron transport chain is going to pass electrons down their structure, their chain, and harvest energy from those electrons. And that energy will then be used to build a proton gradient, just like what we saw during cellular respiration. Okay, let's review. The chlorophyll molecules in the antenna complex in photosystem 2 are going to transmit energy to the reaction center. The reaction center is going to transfer electrons to an electron acceptor molecule. That electron acceptor molecule is one we haven't seen yet. It's called pheophytin. Pheophytin will become reduced when it accepts the electrons from the reaction center. It will then become oxidized as it donates the electrons to the electron transport chain. The electron transport chain is going to pass the electrons along and harvest energy from them. That energy will then be used to pump protons and build a proton gradient. The proton gradient will be allowed to resolve by flowing through the molecule called ATP synthase. and ATP synthase will generate ATP. This sounds very familiar to us, doesn't it? Now let's summarize Photosystem One. In Photosystem One, electrons will also be donated to the electron transport chain. But this time, the electron acceptor molecule receives the electrons not directly from the reaction center, but as they're being passed down the electron transport chain. It looks a little bit different this time. The electron acceptor molecule in Photosystem One is called Ferredoxin. It will turn around and donate those electrons to an electron carrier called NADP+. This electron carrier is different from the NAD+ molecule we saw in mitochondria. The NADP+ electron acceptor will become NADPH, and we will have our product from Photosystem One. Let's review. The antenna complex transmits energy to the reaction center in Photosystem One. The reaction center transfers electrons directly to the electron transport chain. The electron transport chain then donates those electrons to an electron acceptor molecule called Ferredoxin. Ferredoxin then donates those electrons to NADP+, and it generates NADPH. On this slide, we can see two drawings that represent the process that goes on in Photosystem 2 and Photosystem 1. Let's start in the upper left-hand corner. That's the drawing for Photosystem 2. Remember, this is occurring inside the chloroplast organelle, and we're in the interior of the chloroplast in the thylakoid membrane. On the left-hand side of the diagram, you can see a depiction of the energy of the electrons from lower at the bottom to higher at the top. We have a depiction of a photon of light coming in and hitting a chlorophyll molecule in the antenna complex. Remember, that's going to excite an electron in chlorophyll to a higher energy state. That energy will then be transferred to the next chlorophyll molecule. Its electron will get excited. That energy will then be transferred to the next, and then the next, and then the next, until it hits a chlorophyll molecule in the reaction center. And that's what's depicted here. The reaction center chlorophyll is going to transfer the actual electrons to an electron acceptor. That's shown here, and that molecule is called pheophytin. The pheophytin will then donate those electrons to an electron transport chain. The electron transport chain will then pass the electrons along its length and generate a proton gradient with the energy that's harvested. That proton gradient will be allowed to resolve through ATP synthase, and ATP will be produced. In the lower right-hand corner, we have a drawing of the process that goes on in Photosystem 1. Now you'll notice that in this drawing, they've depicted two photons coming in and hitting the antenna complex. Don't worry about that detail. Just understand that photons will be interacting with chlorophyll in the antenna complex. Energy will be transferred around the antenna complex until it hits the reaction center. Electrons will be transferred this time directly into the electron transport chain. It's the electron transport chain that will then hand those electrons to Ferredoxin. And Ferredoxin will hand off the electrons to NADP+, which will be reduced to NADPH. So Photosystem 2 and Photosystem 1 work in conjunction with each other. Photosystem 2 acts first, followed by Photosystem 1. They comprise what are known as the light-dependent reactions of photosynthesis. And if we go back to our sunshine diagram, remember on this diagram they're referred to as light-capturing reactions, but the idea is the same. The light from the sun, along with water, will be used to create both ATP in photosystem 2 and NADPH in photosystem 1. Think about what's happened here. Light energy, again from the sun, is being transformed. It's being converted into a different kind of energy in the light-dependent reactions. It's being transformed into chemical energy, stored energy. That chemical energy will then be used to fuel the Calvin cycle, which is the process that will generate carbohydrate. A couple other notes about photosystem 2. There's a couple of terms that we need to learn that are associated with the events of photosystem 2. Remember, we're inside the chloroplast, and this electron transport chain is sitting in the thylakoid membrane. And just like we saw in the mitochondria, this electron transport chain not only contains these specialized proteins, but it also contains quinones and cytochromes. Remember, those are the molecules that are going to help pass electrons between the complexes in the electron transport chain. Remember, too, that when electrons pass down an electron transport chain, their energy is being stepped down, and that energy is being harvested. The energy that's harvested is then used to pump protons across the thylakoid membrane. And just like we saw in respiration, in photosystem 2, the proton gradient will drive the production of ATP. It will flow through the molecule called ATP synthase, which is also embedded in the thylakoid membrane. And the motion of ATP synthase, the spinning of ATP synthase, will provide the energy to build ATP. The new term for us here is proton motive force. Remember, there is potential energy in an ion gradient. Ion gradients are a way for a cell to temporarily store energy and then turn around and use that energy by allowing the gradient to resolve. The kind of energy that is stored in an ion gradient gets a name. We refer to this as a proton motive force. This, of course, only applies to proton gradients, hydrogen ion gradients. The other term that we need to know is this term chemiosmosis. The term chemiosmosis refers to the coupling of a proton motive force to the production of ATP. So we can now say that chemiosmosis occurs during both cellular respiration and photosynthesis. On this slide, we're looking at a diagram of a region of the thylakoid membrane inside a chloroplast. You can see the piece of membrane over here on the left-hand side of the slide, and it's passing all the way across to the other side of the slide. Now, what we're looking at specifically is photosystem 2, right here, the electron transport chain, right here, and ATP synthase, right here. Let's walk through this diagram together. You can see the photon of light coming in and interacting with chlorophyll in the antenna complex. Remember, that energy will be transferred from chlorophyll to chlorophyll to chlorophyll. until it reaches the reaction center, which they're showing us here. The reaction center will hand the electrons themselves to pheophytin, which will then donate those electrons into the electron transport chain. Here's the electron transport chain here and here, depicted in green again. Notice that there are both quinones, and this happens to be labeled PQ, and cytochromes present. The electron transport chain is harvesting energy from those electrons and using that energy to pump protons from the stroma here into the thylakoid lumen here. We can see a large buildup of hydrogen ions in the thylakoid lumen. Those protons, those hydrogen ions, will be allowed to flow back through ATP synthase into the stroma. That will cause ATP synthase to spin and generate ATP. Light energy is captured during the light-dependent reactions of photosynthesis. It's captured by a pigment, by chlorophyll, and it is converted or transformed into chemical energy in the molecule called ATP. What's critical to remember here is that this ATP that gets produced by photosystem 2 is not going to be used for energy needs in that cell. This ATP is not intended to be used for energy. It's not intended to couple endergonic and exergonic reactions in the cell. That will be handled by the ATP that's produced during cellular respiration. The ATP that's being produced in the chloroplast is just going to be substrate for the Calvin cycle. And so we're going to give the production of ATP this way a new name. Yes, it still occurs with ATP synthase and the proton gradient, the proton motive force. This is an example of chemiosmosis. But because it is driven by light energy, we refer to this as photophosphorylation. So we've seen ATP produced through substrate level phosphorylation. We've seen it produced through oxidative phosphorylation, which, remember, refers to the long chain of oxidation reduction reactions during cellular respiration. And now we've seen ATP produced through the harvesting of light energy. That's photophosphorylation. One other thing to remember about photosystem 2. The electron that gets transferred from the reaction center chlorophyll to pheophytin, it has to be replaced. Remember, that electron came from the reaction center, from the chlorophyll in the reaction center. That chlorophyll has been oxidized. If it's going to be able to receive more electron energy from the antenna complex, we need to replace the electron in the reaction center. That's where water comes in. If we look at the equation for photosynthesis, carbon dioxide plus water plus light energy creates glucose plus oxygen plus water. This water that's on the substrate side of the equation is critical for replacing the electrons that get lost from the reaction center. That water is going to be split into oxygen, electrons, and protons. The equation is shown down here at the bottom of the slide. That water will be broken apart to produce hydrogen ion, electrons, and oxygen gas. This is a very unusual reaction. You don't see this reaction taking place in any other part of the cell. We call it splitting water. and it renews the electrons that are lost from the reaction center.